I'm on Fry, this is experiment 1, what's in the water, and I had sample 35. Did you know, since 2019, more than 320 toxic substances have been found in the United States drinking water, according to the Environmental Working Group, or EWG. Now, introduction. The purpose of this experiment was to determine the unknown compounds that were in the samples that were provided to us by Dr. Dwyer. Now, we use infrared spectroscopy, IR, and nuclear magnetic resonance, which is NMR, to test this chemical to figure out what exactly is in, what chemicals are in this compound. Experimental. Now, using a 368 magnet and an EFT60 NMR spectrometer, that's how we did our NMR tests. First, we prepared in CdCl3, which is deuterated chloroform, and then we had to wipe down the holder and place it in the machine to make sure that no oils were on the vial before we placed it in the machine. After the IR test, we used Thermoscientific Nicolette IS5 and ID7 with a diamond surface. For this, we had to put 3 drops of the chemical substance on a plate and then cover it with an O-ring to ensure that none of it leaked into the machine. After we placed it in the machine, it then proceeded to do the test. Now, what is IR spectroscopy? So this gives us a graph of absorbed light versus frequency or wavelength. The IR region is where the energy required to excite the molecule is. You'll see in the graph in the next couple slides. The separate areas of partial positive and negative charge let a molecule get excited, and then this charge in turn, energy allows it to change the dipole moment in the molecule. Now, what is NMR spectroscopy? So all nuclei are electrically charged and may have a spin. Now, the transfer of energy occurs at a wavelength coincides with radio frequency. Now, this will display the number of peaks of hydrogen atoms. This will allow us to notice how many hydrogen atoms are in the chemical compound and then compare it to other chemicals as well. Now, the results of our IR test. So we have 2 important divs at about 1,700 centimeters and about 3,000 centimeters in here and here. Now, these 2 divs being functional groups of C double bondo and CH, which is singly bonded, are in the sample. Again, our sample is sample 35. 4 different molecule possibilities have C double O and CH singly bonded in their IR graph, which you'll see on the chart next. So here's the chart that we provided in the lab manual that was given to us by Dr. Dwyer. And now here are the 4 chemicals that have these 4 divs, or those 2 divs exclusively. And then we moved on to the NMR tests. And these are the results. So we have 2 lines present, which means there are 1-to-1 ratio, 1 and 1. And then 8 different molecule possibilities have 2 peaks in their NMR graphs. So if we look at the next slide, we can see these are the number of chemicals that have the 2 peaks, and these are the ratios. As you can see, there's only 2 right here that have a 1-to-1 ratio, which is methyl acetate and 1,4-dichlorobutane. Now, if we use our results for the IR and NMR tests together, we can determine that methyl acetate is a compound which is found in sample 35. As we saw earlier, it had 2 peaks from the NMR graph, which are 1-to-1 ratio, as well as a C double O and a CH single bonded found from the IR results. So using the results, we can determine that this is the only chemical possible. Now, discussion, methyl acetate in our drinking water. So some possibilities, or some problems that may occur from having this in our drinking water, is that long-term exposure could lead to irritation of the skin and/or possible with nervous system damage if consumed too much. And then picture here is just an example of what it's used in, and that was by casewayproducts.com. It could also, extensive exposure could lead to dermatitis and/or it's also a solvent in lacquers and paint removers, and inhalation of this product could result in a loss of consciousness or just pass out. It's also found in a lot of nail polish products. And then environmental impacts. It's not toxic to aquatic life, so if it was, spilled in the ocean or anything like that, it wouldn't harm any fish or anything. And generally, it's biodegradable, non-toxic to soil, and it can actually form in food as well. And then these are some of the references that we used.